Antibiotic resistance has emerged as one of the most pressing public health challenges of the 21st century. The rise of so-called superbugs, pathogenic bacteria that have developed resistance to multiple antibiotics, threatens to render some of our most vital medical treatments obsolete. This crisis has been fueled by the overuse and misuse of antibiotics in both human medicine and agriculture, coupled with a significant slowdown in the development of new antibiotics. The golden age of antibiotic discovery, which began in the 1920s with Alexander Fleming's discovery of penicillin, seemed to offer a panacea for bacterial infections. However, bacteria are remarkably adept at evolving mechanisms to evade the effects of these drugs. In the ensuing decades, the pharmaceutical industry managed to stay a step ahead by continually developing new antibiotics, but the pace of innovation has dramatically slowed since the late 20th century. One of the key reasons for this slowdown is economic. Developing a new antibiotic is a costly and lengthy process, often taking over a decade and requiring an investment of over $1 billion. Yet, once an antibiotic is brought to market, the return on investment can be surprisingly low. Unlike drugs for chronic conditions, which patients may take for years, antibiotics are typically prescribed for short courses. Moreover, to preserve the efficacy of new antibiotics and prevent resistance, their use is often restricted, further limiting potential sales. This economic disincentive has led many pharmaceutical companies to abandon antibiotic research altogether, shifting their focus to more profitable areas such as chronic diseases and lifestyle drugs. Despite these challenges, the scientific community is exploring several innovative strategies to combat antibiotic resistance. One promising approach is the discovery of entirely new classes of antibiotics. Traditional methods of antibiotic discovery, which relied heavily on screening soil samples for naturally occurring compounds, have largely been exhausted. However, advances in genomics and synthetic biology are opening up new avenues for drug discovery. By sequencing the genomes of bacteria, scientists can identify genes that produce novel antibiotic compounds. Additionally, synthetic biology techniques allow researchers to engineer bacteria and other organisms to produce these compounds in the lab. Another strategy involves reviving and optimizing older antibiotics that had previously been shelved due to toxicity or other issues. Advances in drug formulation and delivery can help mitigate some of these problems, making these older drugs viable options once again. For example, certain formulations can target the drug more precisely to the site of infection, reducing systemic toxicity. Combination therapies are also being explored as a way to outsmart resistant bacteria. By using two or more antibiotics together, or combining an antibiotic with a compound that inhibits resistance mechanisms, it may be possible to overcome bacterial defenses. This approach requires a deep understanding of the molecular mechanisms of resistance, which has been greatly facilitated by advancements in molecular biology and biochemistry. Beyond developing new antibiotics, there is also a growing emphasis on alternative therapies. Bacteriophage therapy, which uses viruses that specifically infect bacteria, is one such alternative. Bacteriophages or phages are highly specific to their bacterial hosts and can be used to target particular pathogens without harming the beneficial bacteria in the body. Another promising area is the use of antimicrobial peptides, which are part of the innate immune system in many organisms and can kill bacteria by disrupting their cell membranes. In addition to these scientific and medical approaches, addressing antibiotic resistance also requires coordinated policy and public health efforts. This includes stricter regulations on antibiotic use in agriculture, improved infection control practices in healthcare settings, and global surveillance systems to track the spread of resistant bacteria. Public education campaigns are also crucial to reduce the misuse of antibiotics, such as taking antibiotics for viral infections or not completing prescribed courses. Ultimately, the fight against superbugs will require a multifaceted approach that combines scientific innovation with robust public health strategies. While the challenges are formidable, the potential consequences of inaction are dire. Without effective antibiotics, routine surgeries, cancer treatments, and even minor injuries could become life-threatening. By investing in research, fostering collaboration between the public and private sectors, and promoting responsible antibiotic use, we can develop the tools needed to stay ahead in the battle against resistant bacteria. The resilience and adaptability of bacteria are undeniable, but so too is the ingenuity and determination of the scientific community. Comment with your favorite fact.